Okay, good evening, everyone. Welcome to tonight's Ahead of Cancer lecture series sponsored by the Allegheny Health Network's Cancer Institute, and thank you for joining us. My name is Michelle Studnicki, and I'm a project coordinator for the Cancer Institute. Before we begin, I'd like to go over a few housekeeping items. Number one, please feel free to submit questions via the Zoom chat or the Q&A feature throughout the presentation. We'll address as many as time allows at the very end of the lecture. Number two, for this webinar, participants will be muted. However, we encourage your participation throughout the chat by using through the chat function. Tonight, we are delighted to welcome Dr. Nathan Bahary, who will be presenting on clinical trials, what, when, and why. A little background about Dr. Bahary. Dr. Bahary is the Division Chief of Medical Oncology and the Director of Clinical Research at the Allegheny Health Network's Cancer Institute. Board certified in clinical oncology, Dr. Bahary received his medical degree from Cornell University Medical College in New York, Cornell Medical College, and his PhD from the Rockefeller University in New York. He completed his residency at Beth Israel Deaconess Medical Center in Boston and a research postdoctoral fellowship at Harvard Medical School in Boston. He is a member of the ECOG GI Committee and the NCI Pancreatic Force. Dr. Bahary's research interests include identifying novel genes and pathways underlying normal gastrointestinal development and cancer to both design rational-based disease markers for early detection of gastrointestinal cancers, as well as rational-directed therapies for their treatment. Dr. Bahary joined the Allegheny Health Network Cancer Institute in 2021 as the Division Chief for Medical Oncology and the Director of the Allegheny Health Network Cancer Institute's Clinical Trials Program. Welcome, Dr. Bahary.
Thank you very much, Michelle, for that very kind introduction. I promised to get everybody out in time for the Thursday night football game on Amazon.Prime. You can choose the Rams or San Francisco, whichever is in the Steelers' best interest. How is that? So just conflicts of interest first. I'm a consultant to a number of companies, and all of that is not personal to me. It's all to get research support for the clinical trials. I will not be discussing off-label drug usage outside of FDA restrictions. So here is the entire talk in one slide. The entire reason that we are here and the entire reason the clinical trials program exists is to advance cancer care. And they're really the means. Trials are really the means to identify the next generation treatments. And we want to bring them to cancer patients. And so we can't do everything all at once, but by incrementally improving care, we can make a huge difference in people's lives. There's different phases of a clinical trial. There's preclinical development, where you take things that have been rationally found, and everything comes from the basic science world, and they throw it in test tubes or petri dishes and eventually into animal models of disease. And then they do the first human trials, and it uses only a few small doses. It's called a phase 0 trial, and it's really to see how the drugs behave, to make certain people don't get really sick with it from the way they're producing it. And then we get to phase 1. And the goal of that, lots of people, these are new drugs, new treatments, new treatment combinations, and if they're safe for people. So it's really a dose escalation side effect trial. What we are really trying to do is understand what's the maximum tolerated dose, what side effects, and then you can go back, and these are usually pretty inclusive of many different types of cancers. And then the idea is to go back and see, did we see any utility in them in whatever way we're looking at it. And then we can go on to phase 2, which we might say is just going to be ovarian cancers or brain cancers or lung cancers, or maybe it's multiple GI cancers, or it's all cancers with a certain change in it. But it tells you more about how safe the treatment is and how well it works. And so it can be a phase 2 study, can be what's called a single arm study. That is, everybody gets the intervention, or a small randomized one, where we don't quite have statistical power, and we're not going to really go into that, to get like FDA approval, to prove beyond a reasonable doubt it's better than the standard of care. But it's another path on the way to a large phase 3 trial. And these are randomized studies, and you use them to compare a new treatment with the standard treatment. And we want to know if the treatment is better. Do people live longer? Perhaps they have a better quality of life. Perhaps they have less side effects than the drugs. So each of these trials can have different endpoints. We could be looking at different things, but each of them are vitally important. So let's talk about the first randomized clinical trial. I always like talking about this when I give talks on this subject, and it was back in 1747. Dr. James Lind, he was on a ship going across the ocean to the New World, and a lot of sailors got scurvy, and it causes swollen, bleeding gums, blue spots on the skin, and people actually can die of it. And it all comes from a lack of vitamin C. And it turns out that us and hamsters are the only entities which don't make their own vitamin C. But they didn't know that at the time. So he had took 12 patients. Two were ordered to get a quart of cider every day. I've got a feeling it was kind of a spiked cider. They got some elixir vitriol, doesn't say what those are. Two spoonfuls of vinegar 3 times a day. Two of the worst patients they actually put on seawater. Obviously, that didn't really help. And two others got 2 oranges and a lemon. And the two remaining patients were really done amazingly, and they had visible side effects that were immediate from the use of the oranges and lemons. One of those who had been taken off duty was in 6 days was fit for duty. And since it started on May 20th, Clinical Trials Day is held of May 20th every year to commemorate the day he began this trial. And yet he also opened up the can of worms known as insurance coverage. So he recommended the use of oranges and lemons, but the Navy actually rejected it because they were too expensive. So it was nearly 50 years before they made lemon juice a compulsory part. And then it was lime juice because it was significantly cheaper when they figured out they were essentially the same things. So what about lessons? In the grade, we're going to have lesson number one, trials can come in many forms. Although we're going to be talking mostly about clinical trials that we do with cancer patients with new drugs or so, there are other types of ones. For instance, looking at whether cigarette smoking was the leading cause of cancer. There were population studies. People looked at the population and they noticed that as people began to smoke more, they got lung cancer incidence rose. As Virginia Slims, you've come a long way, baby, came. That was the when I was young, their tagline, and skyrocketing risks of lung cancer in women. Multiple studies suggested cohort studies. So what they do is they compare people. They say who are the people who smoke and don't smoke, who live in the same area, might be exposed to the same thing. And they concluded in this study that there was no reasonable doubt that smoking led to lung cancer. So these are kind of population studies. They did animal experimentation. You take tar and stuff and inject it into mice, and guess what? They got cancer. They looked at the lung lining and they said, wow, the things that clear out all our mucus aren't really working when people are smoking. And then in the 50s into the 60s, they found cancer-causing chemicals in cigarette smoke. And so it became, it was somehow people obfuscated it for a very long time. But it was pretty clear at that point that the two were linked. And different types of trials can lead to the same conclusion. So the lesson here I want to show you is it took a long time to do this. The first treatments for lung cancer was surgery, then radiation, and the radiation then was quite crude. We gave chemotherapy in the 1980s, combinations of chemotherapies in the 1990s as I was training. And this was one of the biggest studies. It was 4 different combinations of doublets, meaning 2 chemotherapy agents. And, you can see like they were all the same. They have different side effects. You could choose between them. But you can see that we really didn't do a whole lot, right? People were still dying. Most people were dead in 18 to 24 months. Half the people were dead in about 8 months. And nobody was really alive after 3 years. So then came targeted therapy, as we'll talk about. Then we're adding it to chemotherapy, further targeted therapy, and now we've got immune therapy added to chemotherapy. And so this took literally 50 years and is still ongoing. And so, people say, why isn't it done? Well, as we're going to show you through the talk, that all progress is somewhat slow. So what is this targeted therapy? We're going to talk a bit about that. And basically, the lesson that I want people to take away from here is all advances stem from basic science. And just in case you don't know, DNA is the biological unit of data storage. It's what's inherited from parents to children and makes each of us an individual. And different combinations of these four values of what the basis can be is the blueprint for life. And so a gene, a string of these, is the basic unit of hereditary passed from patient to parent to child. And mutations, changes in that DNA can activate a gene, that is, turn it on, and that will make it to cause growth. Or it can be deactivated, like when people lose the hemoglobin genes in sickle cell disease and facilitating a disease. So driver mutations, we call them that, describes changes in the DNA that causes cancer cells to grow and multiply. So this is a type of lung cancer, driver mutations, and non-small cell lung cancer. And think of it as analogous to driving your car, that these driver changes are in the DNA facilitate cancer growth. So I like to explain to patients, this is if I took a broomstick, put it on the throttle and tied it down, and the car is going to go awfully quick and be difficult to stop. And so what we found that actually is that different genes, different units of heredity were actually altered. This gene, don't worry about what they are, but one's called KRAS, EGFR, HER2, ALK. We'll talk about these things. But when these things were changed, it actually drove the cancer. So if you took this, put it in an animal, you could recapitulate the lung cancer. And then you get to the basic science. How do we turn these genes off? So the first thing was in 2009. A gene called EGFR was mutated, altered, turned on, broomstick on the cancer throttle. And it turned out that it was effective, and another one called erlotinib was also effective. There are these ones called ALK, and that they were altered, and there's a gene called crizotinib. And then found out that this gene also was in ROS1. It's a different gene. But then they proved in 2014 you were better targeting these tumors, and that means we had to start checking everybody for these changes in the tumor DNA. And then we found that it was all really nice, but cancers grew resistant to it. They found newer drugs that can get past that resistance. And now they have drugs that can target a whole array of these things, and all of them are affected, such that we've actually completely changed the landscape in non-small cell lung cancer by looking for these targeted therapies. And each one had to be tested in a trial in order to prove its effectiveness before we could bring it out. So we're going to talk about a survival curve. It's called Kaplan-Meier after the two statisticians, and it's a randomized, it's a trial survival curve, and it shows the probability of a patient surviving or not experiencing an event of interest. For instance, if you were studying anti-nausea medications, it may not be survival, but how many times you had nausea with the chemotherapy with a different drug. And so they're randomly assigned to different treatment groups, and a key tool for assessing the efficacy, how well a new treatment works. So up and down on the y-axis will show you is the probability of survival or not experience an event, and from left to right is the time elapsed from the start of a trial. So this is targeted therapy. And down here are chemotherapy and immune therapies were just becoming on the market. You can see, again, we call it the median survival. Half the people were dead in about 4 to 5 months with the chemotherapy or something else. And yet you can see here, half the people were living more than a year and a half. But what was amazing is that this is tail to the curve. And some 10, and now we're with newer drugs, up to about 20% of these people live 5, 7, 10 years. And many of these people tend to be non-smokers who are younger, actually, and it's really been a game changer for them. So we'll look at these survival curves, and we'll look at a few more, and the idea is to see the number of people alive in this case over time, and you can clearly see there's a difference between these sets of lines, and the top one is better. So another lesson to learn, that you can do a trial in one disease, and you can probably expand it to include multiple similar mutations or profiles in other diseases. So for instance, the EGFR, which we talked about in lung cancer, can also be found in colon cancer. ALK in a bunch of diseases, HER2 in a bunch of diseases, KRAS in a bunch of diseases. All of these now, we actually, when we get patients with cancer, we test for all of these genes now. And it's rather amazing that we can test for all of these in a couple-week period of time because the very first human genome took $10 billion and about a decade. And we can do this for a few $1,000s in a week and a half or 2 weeks. And so then we can come back and decide if we can use special targeted treatments. So to give you an idea, this was a young patient I had with pancreas cancer, and it turned out he had an ALK mutation. This is actually a translocation, two pieces of DNA stuck together, turning ALK on. In the left, you can see we're pointing arrows to it. You wouldn't know, but that's a spleen, that's a liver, here are his kidneys. This is a lymph node here, and these are other lymph nodes. And he had these up and down his spine, causing terrible pain because that's where the nerves came out. And after just a few months on it, they disappeared. And he went on to live a great number of years and had a wonderful quality of life. Biliary cancers, bile ducts, they're the things that carry bile from the gallbladder, from the liver, that help dissolve food in your intestines. And it turns out this is just shows you there's many different kinds of these, but there's many recurrent changes and in all of these different genes. And if you take a look now, if you take a look at these and add them to the most common ones, up to 40% of people who have biliary cancers actually can have a targeted therapy. And so it's become standard of care to try to find these to improve people's overall survival. So here's a gentleman who's shortly after a drug called pemigatinib, which targets a gene called FGFR2. He had treatment-resistant bile duct cancer. He was headed actually to hospice when this came out. And this is just a blood test that shows you how active the cancer is. And you can see it was decreasing nicely. And he went on to live close to a decade on this drug. So what are the pitfalls of some of this? Temper your enthusiasm. So I'm going to give you a couple of examples. People come to me and say, I read this study and people had incredible response rates and people did really well and I want this drug. And, they're pushing for it. And why are we even going to test it? Well, there's certain things that biases that go into a study. One is like ascertainment bias. And the easiest example to understand this, if I went into nursing homes and said, does being in a nursing home cause you to have prostate cancer? You might find very little prostate cancer because in nursing homes, they're mostly women, right, who outlive men. So where you choose to do this and how you choose to do it can introduce bias. Stage issues. When we were looking at screening for prostate cancer, people said it increased survival by 7 or 8 years. Well, actually, what you did is you found the cancer 7 or 8 years earlier. They didn't live much longer. Now, there's subgroups, temper that. I mean, talk to your urologist before you say you don't want a PSA or test or so. But, it became far more nuanced. And so that gets into this idea of lead time bias, right? We can find it earlier or in different populations and goose the results accidentally. So here was a trial when I was previous at a different institution prior to coming to AHN that we participated in, and it was gemcitabine and a drug called bevacizumab or Avastin, and the thought was this would starve tumors of blood. It's a blood vessel inhibitor. And the response rate was higher than anybody had seen before. Median survival was like twice as high as ever been seen before. And then this was a study that was mostly done at the University of Chicago. We got to put a couple patients on it through a group, but about 80–90% of them were all local to the University of Chicago. Well, the University of Chicago happens to be, I grew up right near there, Hyde Park, with a lot of people who are quite a lot of people have a lot of education. They're teaching at the University of Chicago. It's a different group of people, such that when they actually took 602 patients and randomized people to the combination doublet, this drug plus that versus placebo, there's two lines under here. You might not be able to tell, but believe me, there's a blue one and a yellow one, and the survival was exactly the same, and the response rates were actually pretty low, which is what we usually see. So this is where we can hope for more than's actually there. And we can't try to cut corners. We don't do our patients, we don't do medicine justice when we try to cut corners and just think we know the answer. Here's another one that I was just recently involved with. The actual phase one, this was didn't even go to phase two. Phase one was done with 20 patients in Europe, and these are pancreatic cancer patients who almost never have any response. And they found these people who had almost complete responses, and they lived 2 or 3 times longer than ever had seen before, in combination with chemotherapy. So that's me. I was the one of the larger accruals to the trial worldwide. And you can see this drug now had a name with FOLFOX, with this chemotherapy versus the chemotherapy alone. And there's two survival curves in here. And they're exactly the same. There's absolutely no difference. And, I know that when we were trying to administer this, it was so hard. This ended up like on the front page of the Wall Street Journal. People wanted to invest in it. Patients were lobbying the leadership of where I was, insisting that their family member get the drug. We couldn't randomize them to it. And if we did that, we would have been in a lot of trouble, right? Because we would have never found out, believe it or not, it doesn't work. So what I'd like to do is shift from drugs that are like standard chemotherapy to the immune system, because that is a really important thing. And so the immune system is very important in cancers, and we're learning more. So for instance, these DNA genes get damaged due to smoking, light, UV light causes melanoma or other problems. And these changes in the DNA can be seen by the immune system, and you can actually get an immune response to it. And so we call the antigens are things the immune system sees. Neoantigens are new antigens, and they're processed in the immune system, and actually they can be seen. And it turns out there are immune checkpoints, and they're designed to suppress the immune system. So you can get sick with the flu, with fevers and everything, because your immune system is fighting the flu, except you want it to eventually stop. You don't want to feel like that forever. And so there are these checkpoints. And what it turns out is that cancer cells take over these checkpoints. The immune system can no longer target them, so let cancer cells thrive and grow. And just in the last 8 years, there were these checkpoint inhibitors that actually won the Nobel Prize. This is one of them called pembrolizumab, and it inhibits something called PD-1, programmed death ligand 1. And it turns out if you take a look at this, these patients who had immune therapy versus a different immune therapy, these are two immune therapies being compared against each other. Remember that non-small cell lung cancer, when everybody, but we have about 10% of people living at 4 or 5 years. Now we've got people that was targeted therapy, and no one was really out there with chemotherapy. Now we have 40% of people alive at 5 years. It's really been an amazing difference or so. And it shows you that this immune therapy is better than that immune therapy. And yes, people are trying to combine them in order to improve it. And that'll be another study. So a lesson here is chance favors the prepared mind. When these checkpoints came out, everybody said, we're going to try it for everything. And so they tried it in a bunch of different cancers. So this was an early phase study. This drug was called MDX 1106 before it was Merck Sharp and Dohme at that time, before it got the name pembrolizumab. And it was mostly studied in melanoma, which we had a reason to believe that it might respond to immune therapies, as well as kidney cancers, bladder cancers, because they're harmed by smoking, and UV light harms melanomas. But they threw in some other things, and they had 14 patients with colorectal cancer, CRC. In this other one, they had 19 out of 296. And if you add those two up, you get 33, and only one of those 33s had a response for the colorectal cancer, but it was a complete response. They never recurred. And it turns out this patient had hereditary syndrome called Lynch, and it's a DNA repair problem. So what happens is when we harm our DNA by whatever means, we can repair it. And it notices there's changes in between DNA, and they're called mismatches, and it will repair it. So we call it the mismatch repair system. And so they have a problem with this. So they accumulate incredible numbers of mutations. And so we talk about liquid tumors like lymphomas, leukemias, pediatrics don't have many. These are kind of sporadic ones. mismatch repair proficient, meaning they don't have this, colon cancers are here. But look at this, mismatch repair colon cancers are in here, and they have thousands. And every time you have one, you have a much higher chance of the immune system seeing it, such that if you take a look now at patients when they had microsatellite high, mismatch repair deficient metastatic colorectal cancer. And we ask if we give them immune therapy, which is far easier than chemotherapy, the people with immune therapy have progressed far less, this is a huge difference, and survive much longer. And so actually, if you have this type of colon cancer, and we now check every cancer for it, and I'll show you why, that actually immune therapy is now. So they had to do a very large trial, and we had to do immune therapy versus chemotherapy. And yes, people got randomized to the chemotherapy, but we would never have found out if this worked so well without this trial. So the next thing that came up with it was called a basket trial. As you look at anything, gallbladders, cholangiomas, endometrial or uterine, gastric cancers, esophageal cancers, pancreas, prostate, thyroid, any of these that had the same deficient mismatch repair. So they screened all of these tumors. If you had this in your tumor, it was you actually could get on this drug. And you can see this is what's called a waterfall plot, meaning you just see how deep the responses are. People had complete responses. Many of these partial responses were actually contained over years. And so this drug got an agnostic, meaning you could use it for any tumor that had this DNA look to it. So it's another thing that we do when we look at the DNA and we look for this mismatch repair. And this is a kind of basket trial. So this was not randomized, but it was just to look to historical controls. Normally, all of these tumors, people die very quickly. And to have this kind of response, you don't even have to randomize to it. So to give you an idea, this was a cholangiocarcinoma, a bile duct tumor that I had, a 58-year-old gentleman who had this, failed all available chemotherapy. I was just coming out with this ability to look for this. And I said, let's try it. I got some free ones from a Foundation Medicine. And we placed them on hospice, thinking we wouldn't find anything on. I was at a meeting when they called me up on a Saturday night at 11 o'clock at this meeting to tell me that he had lost this protein, this MLH1, which causes mismatch repair. We put him on, we got this trial opened really quickly that I talked to you about. His tumor normalized, 3 months, no evidence of disease, remains on pembrolizumab since 2015. And we'd like him to stop, but he's a little bit too scared. But we found that you could probably stop in a couple of years and you will be cured at that point. So here's somebody who you literally grabbed from the jaws of death to a cure by something new that came out of a trial. How about looking at these in non-small cell lung cancers? Why would we do that? Well, remember, lung cancers are caused by smoking. Smoking causes changes in DNA. And so they looked at something called, in this trial, this was the same trial that we talked about with survival with immune therapy. But what they did is they looked at a marker, this PD-L1 test. So it's something on the membrane that lets the immune system kind of see it. And so this has a lot of brown on there, right? All these are tumor cells. And that we say it's a high PD-L1 expression, and this is low. And if you take a look at the people who were low, they received very little benefit. And the people who had a lot of it, more than 50% of their cells, actually had incredible benefit and would go on to many of these people to live for many years or so. So this drug alone, this immune therapy, in a trial, we could look at a predictor of response and then come back and try to have a better idea of when to use it. So this is looking at chemotherapy plus the thing. So if immune therapy is good, can we add it to chemotherapy? And so these are different proportions of the expression of the PD-L1. And it turns out, whereas, remember, if it was very low expression of this marker, right, on the cells, we didn't get any benefit from the pembrolizumab, the immune therapy. Actually, chemotherapy plus the immune therapy even helped those. So there's some interaction between chemotherapy and the immune therapy that benefits it. But if you really look out here, if you have a lot of it, it's huge. right? The combination. And most of these placebos would be dead in a few years, and these pembrolizumabs about half are alive in 5 years or so. So this is just an amazing thing that says, what are you going to do? You can take one thing and try to add it to another, but again, it requires a good clinical trial to be able to do it. So another lesson is, can we move it to earlier stages? There's a lot of evidence to suggest that it's harder to treat cancers in later stage than earlier stage. So neoadjuvant means prior to surgery. So in rectal cancer, what we did was we originally would take it out and then give chemotherapy and radiation therapy afterwards. And about 20 years ago, we came to realize that actually if you gave chemotherapy and or radiation first, and then did surgery, so sort of these white boxes, people did much better because they were much healthier to be able to do it. So we started looking at this mismatch repair and rectal cancers that actually could have surgery. And what they gave is this immune therapy. It's just like the pembrolizumab you gave for a number of months. They took a look. If they had residual disease, they would go on to get chemotherapy, radiation therapy, a very large surgery. Most of these patients would have had to have what's called an abdominal peritoneal resection, which means removing the bottom of the rectum, the anus, and having a ostomy, a bag, for the rest of your life. And it turned out every single one had a clinical complete response without recurrence. And so we now know that we need to test for this in rectal cancers. And now there's studies to see if the same thing is true in colon cancers, not just down in the rectum. But this became because of these trials. And, people looked at how you do it and said, can you use double combinations of two different immune therapies? Those are ongoing to even perhaps make it more stable and more long term. And so if it works in rectal cancer, remember we said if it works in one thing, maybe it'll work in another. Decided to give this nivolumab, another one of these immune therapies, and plus chemotherapy prior to it. Surgery, and if you gave chemotherapy alone before surgery, which we know is better in many of these tumors, not all lung cancers, but the ones that they looked at here. And if you gave the immune therapy plus chemotherapy, a larger proportion of these people were cured. And so this has now become the standard of care, moving the chemotherapy before the surgery, adding immune therapy to it. And there's different variations on this theme, which immune therapy for how long, but the general principle remains that what we learned in one, we could take to another one. And now for melanoma, melanoma is one of the most immune treatable diseases. It has a very high rate, even in stage 4, when it had like a 3% survival past a couple of years. Now 50% or so are actually living 5 years, even with stage 4, meaning metastatic spread to other organs. And this looked like if we just took it out and then gave people the immune therapy, it's been proven if you gave it to them before and after the immune therapy, that they did much better. So now you're curing another 25 or 30%. So again, what we learn in one disease can be taken to another with a well-designed clinical trial. I'm going to move to something called HER2. It's one of those proteins we told you about. It's something that can be altered. It sits on the cancer cells, and it helps regulate their cell growth. And we mentioned it earlier with non-small cell lung cancer, and we'll try to broaden it a little bit. And historically, if you have too much of this protein, this receptor, is associated with an increased risk of disease recurrence and significantly worse prognosis, especially in breast cancer and stomach cancers. And so these new drugs came out, and it turned out that if you had a lot of HER2, remember we had a lot of that PD-L1, that brown stuff that was on the cells when we investigated it. If you have a lot of HER2, you also would get a lot of benefit from using a targeted agent against this. And now there are multiple drugs that can target it. But we have that. Now the question came up, what do you do, right, to try to improve it? And basic biology, remember I said everything derives from basic science. The thought was to take an antibody. So this is something that's been engineered to look like an antibody which fights infections, but it's actually binds this HER2, and we know that if we use it alone in a drug called like trastuzumab, it can help. But eventually, what happens is you develop resistance. So they said, can we create an antibody drug conjugate? So there's these topoisomerase inhibitors, irinotecan, topotecan. They're used in a lot of different diseases. The idea is, can you link them in many places to this protein and then deliver it? And so you have a lot more drug being delivered directly to the cancer. And there's even what's called a bystander killing effect. So it doesn't have to bind to the cancer, it brings the drug really close to the cancer. And so it's really been incredible. And it turns out that it's effective in breast and stomach cancers after failure of the other targeted agents with high HER2 expression. So this is like that picture that we showed you with all the brown stuff on it. Now, what happens if you don't have a lot? Well, it's been studied in breast cancer because a large proportion of breast cancers actually have low HER2 expression. And so they looked at it in low HER2 expression and they randomized people to the drug and not, and they got accelerated approval for it in breast cancer for low HER2, but also lung cancers and non-small cell lung cancers that have a lot of it. So it was a tumor agnostic approval after a bunch of studies, and these names don't have to mean much, but you can look them up if you feel like it, that this is a HER2-directed therapy with an antibody drug conjugate, a new class of compounds that we studied that have revolutionized treatments of breast cancer because it can be used in low and in every cancer that is highly expressing it from the beginning. So again, like we talked about, tumor agnostic, meaning that it can come from any cancer. And now, along with all these other genes and the mismatch repair, we now have to test for HER2 as well. But it's a great thing to do because we know that we're helping people when we find these. So what is the lesson of this clinical trial scenario? The trials are not always linear. We have preclinical basic science in animal models, right? Bring it to a phase 1, we see toxicity side effects. They brought it into phase 2 in breast and stomach. Breast and stomach cancers made it to phase 3. Then it actually, I should put another arrow because they went back saying, what happens when these tumors are resistant? Take out resistant tumors. So people were willing to have biopsies, right? It's not going to help them, but they were willing to help the us be able to change the medicine model. And it went back to the basic science, back to phase 1 with this HER2 and HER2. And it turned out that it worked in colorectal and lung cancers, and it can be used in any tumor. And again, it was so striking that it managed to get approval just in a phase 2 because other things did not help in these scenarios when you had a HER2-directed therapy. And so you can see that this is a constant back and forth and intersection between basic science and tests and models, right? They do randomized studies in mice and other animal models, and then have to bring it back through in order to bring it into people. And just so that you know the cost of this and why is it not just done willy-nilly, to bring a drug from the preclinical all the way through to getting approval by the FDA can be between $1 and $200 million. And I'm not saying that that justifies high prices, but there is a large price for this. So you have to do these trials. You have to be somewhat exacting about how you collect the data and making certain that entry criteria are met. If you're going to do a randomized study, this phase 3, you better make certain the control group and the treatment group are the same, right? And that you also include as much as possible uh, races and ethnic minorities, right, who are not in there. Otherwise, we won't know how well it works with them. And also worldwide, if we can, to understand really in the real world, how does this work? Not at the University of Chicago, when you've got lots of people who are have advanced degrees and can dig these things out, and we'll get to it earlier. But in the real world, how do people, real world evidence, actually um, benefit from this? So what about rare diseases in clinical trials? This comes up I've got a rare disease. What do I do? So you can get multi-tumor approvals. We take rare diseases and we look at that PDL1. We look for mismatch repair, HER2 agents, NTRX, NTRK, excuse me, NRG translocations. We talked about others, ROS and BRAF. We look for all of those things. We have multi-tumor approvals. And the NCI has an initiative, a rare tumor program. So some people will take things for rare tumors, such as that's one of the things with HER2 low breast cancer that I may not be rare, but it's rare that there's anything that treats it well. And there's initiatives, money set aside to test these things. And then there's pathway specific ones like KRAS. And so until about a decade ago, or maybe even 6 or 7 years ago, the mantra, there was a review that I actually helped write, you could find it. It was KRAS kind of the undruggable target, right? Because it was going to be so hard to target this gene. And it turns out that with AI, right, with this incredible use of computer power, they were able to predict the three-dimensional structure of this and then synthesize something in the lab that would actually target it. So they didn't have to try to do it by crystals. They didn't have to do odd things. They're actually able to do it mostly in vivo, and it worked. And these drugs are making their way through clinical trials. And some foundations are dedicated to specific rare diseases. So one disease that a number of us are interested in is an appendiceal cancer. There's not many cases. Basically, it's a disease of the appendix, tends to spread to the intestinal surfaces. Often the treatment, it's rather refractory to chemotherapy. What we do is surgeons go in a very long 12, 13, 14, 15-hour operation, remove every little dot of cancer on the outside of the intestines, and then put hot chemotherapy directly on top. And it turns out some of these neoplasms, these cancers, have these KRAS and GNAS mutations. And it turns out there was a group in California that had a very large personalized trial. They were doing different DNA tests, and they said if you had, for instance, this mutation, we would give you this chemotherapy, and most of them didn't work. But at the time, they thought there was something called CCND1. Don't worry about what the gene is, but it was amplified, meaning there were multiple copies. The cancer had multiple copies. That this was going to be a biomarker. It was going to be a marker for response, just like PDL1 is to immune therapy to palbociclib. Now that was later disproved, but this one patient got on a personalized trial, got on palbociclib, and had stable disease for about the last 5 years. And so this group went back and took a look at a drug that can perhaps inhibit this, the palbociclib, and they had 16 patients, 13 of which were appendiceal. And it's very hard to see these tumors on scans, but they saw the serum markers. Remember, we talked about that CA19-9, the activity. 80% of them were stable, 38% came down. A number of people had stable disease for an extended periods of time. But interestingly, some people succumbed to the disease, even though they didn't have mutations to it. It was not quite clear. And some people responded well to it despite having resistance mutations. So you don't know exactly. And just to give you an idea of how hard these trials are, they started in 2016 and finished enrollment in 2022 and got reported just a couple of months ago. So, the question is, again, chance favors the prepared mind. Lots of us want to try to use this for these patients, but you have to, hopefully we'll get some further trials as well. And hopefully we can collect the data if people get it outside of a trial so we can find out if it really works. So what about biological therapies, the whole new future? So the idea is that, as I told you about the immune system really being important, that future trials really have to incorporate these new classes of drugs. And so you take a patient's tumor, and in that tumor, if you look, there are immune cells called T cells that actually can fight the cancer. So you can remove them from the patient. You could put them in a dish and find these infiltrating lymphocytes. We call them TILs. And what they are is, if you saw the tumor, you see there's all these white cells, all these lymphocytes, all these immune cells right next to it, right? And a wave, like they're getting ready to try to kill it, but the cancer won't let it. So what they do is you isolate them, then you can expand them in a bioreactor or a machine, and then what you do is you give them back to the patient. And so this is the more of these treatments are coming online. And then there's ones where you can take the T cells out of people, put them in and modify them. So every immune system, remember we said, has on its surface when we were talking about this, something that recognizes a tumor or recognizes the bacteria or recognizes the virus to get rid of it. And what you do is you put into it a modified receptor thing on the surface that actually targets the cancer. And so you create billions or trillions of copies of this that can fight the cancer, and you can actually put it back in the patient or so. And in a number of cases, especially in pediatric cancers, adult leukemias, certain adult lymphomas, you've taken people from the jaws of death, literally with nothing else, who've gone through this, and they're now living 2, 3, 4 years. And you say, are they cured? Well, we have to wait longer because these have only been around for about 3 or 4 years. Ask me in 10 years what the 14-year survival is. But they're looking great, and the cancers haven't come back. And now this works very well in certain types of tumors. Doesn't work particularly well in the solid tumors yet. People are trying to work on it because they're very different, the cancer cells in solid tumors, from like lymphomas, leukemias, which is somewhat monotonous. But there's a lot of people working on this part of it where you can get a whole bunch of different white cells to be able to attack the cancer. And there's been some wonderful preliminary evidence that's being at meetings that is being provided. So here's one. This is advanced melanoma. And these are patients who, we told you about half the people can be cured with advanced melanoma, but that means the other half, or 60%, are actually dying it. Maybe 65% are dying of the melanoma eventually. So the question is, what do you do afterwards? You give them a different immune therapy, and some of these people would live a few years. There was a couple of people who would be cured by it, but not many. And what they did is took out the melanoma. They did this TIL, right? They isolated the T cells in the around surrounding the melanoma, gave them back to people, and now 25% of the people who are destined to die are now cured. And this is actually now commercialized. We were the first institution to open it up. And the second we had the second patient nationwide on it, who's doing remarkably well, actually. And we weren't first by about 5 days, but we're very proud of the fact that we were able to take a trial and then bring the commercialized aspect of it to our patients' benefit very quickly. So there's multiple research trials. There are immune therapies, cell therapies, combination therapies that are in all sorts of active clinical trials here, mostly at West Penn, because that's where a lot of the immune therapy is going on. The intensive ones like the TILs and CAR-Ts, you can get regular immune therapies anywhere through our system. But there are many others in startup, and we're very excited to see some of the responses in these tumors when we didn't have activity or hope for people before. Now, this is a very interesting trial, and it is being spearheaded by Albert and Vera Donnenberg, who are basic scientists. They're PhDs here, along with David Bartlett. And the idea is to harvest. There turns out this is fluid. Sorry about that for those, if you haven't eaten yet, that comes from the lung and people who have fluid in the lung. There's cancer cells through this. And along with the cancer cells, when we remove it, there's these white cells. These are all these cells. And so what they do is they take it out and they grow it on the machine. This is the machine. It's called the maternal prodigy machine. And don't worry about how you select it, but they take a number of these, they expand it hundreds and thousands of fold. And the idea is that all of these cells can be given back to the patient. And there's a very large Department of Defense trial that's going to be opening for this in breast cancer and pleural fluid and lung cancers and pleural fluid probably within the next year. We have to get FDA approval for all of this. But it just shows you how you can take some of the bench stuff, come up with a new idea. No one had thought about going into this fluid for it. They were taking it from the tumor, isolating the cells, growing them on a machine not designed for it, but can be rigged for it, and now it's going to go into clinical trials. So there's different immune therapies. There's vaccines. There's a number of vaccines being tested. Cytokines are things that are given off that can fight cancers, small molecules. The things that we were talking about are adoptive cell transfer. The PD-1 inhibitors like pembrolizumab, dostarlimab are immune checkpoint inhibitors. Dave Bartlett and a number of other people are working on oncolytic virus therapies as well. So this is a virus, they can get into it and specifically try to kill the cancer cells or activate them or work with them or have a preferential for killing them. Just like these, your immune system can get into virally infected cells and kill those, and that's why you get over the flu. So what kind of trials are there? Remember, we talked about basket trials, multiple diseases with the same type of intervention, immune therapy, a targeted therapy. There's sometimes what are called umbrella trials, where what you do is you have a single disease and you have 3 or 4 different interventions. People get randomized to intervention A, could be chemo, chemo plus immune therapy, targeted therapy plus immune therapy, or other types of therapies. So a single disease, sorry, here we go. I'll get there. Going the wrong way. And there's also what are called platform trials. And these are incredibly complicated because people are getting standard of care. Some of the patients are getting intervention 1. Some are getting intervention 2. Sometimes science comes up and there's a whole new thing that's been found, right? Basic science. And so there's a new intervention. This intervention actually doesn't look like it's working, so it's dropped. And later on, a new one is. You do an interim analysis and say, what's working? Well, this is not working, we drop it, but this one might be, and you introduce it. So you're constantly trying to tweak this and see which one of these are better. There's a number of these kind of platform trials being done across the nation, including at Allegheny Health Network. So what are methods to improve the efficiency? This is a very long process. Seems it can take a decade to get something approved. Sometimes you can do it quicker. N of 1 clinical trials, if we think that there's something specific about your tumor, like, I had a patient with an NRG translocated cancer, and we were able to get her a drug that was approved only last week, but we got it for her 2 years ago as a trial, and she responded beautifully to it and is still on it. And that's kind of the expanded access program. Sometimes we treat people off-label. Label is what the FDA says it's right, it's supposed to be used for. And a lot of these decentralized trials, right? We have to get consortiums together. As I told you about getting a little cocky and taking your phase 2 trial and saying it's good for everybody, we really have to prove it works in the real world. So what's something very interesting? I'm going to end on a couple things that we're doing here at AHN. One of this is called the Moonshot Program. And the idea is to define novel markers of cancer development, predict response, predict toxicity. So we ask everybody who comes in with cancer, and this is only through September of 2024. As of last month, we're about 3,000 blood samples, enrolled patients. And what we ask is before you start the chemotherapy or whatever it is, you start your surgery, you give a little bit of tumor and blood. So it doesn't require anything. It's just when you're getting a blood draw, we take a little bit more. And then you get monthly blood draws, and it decreases over follow-up, and we correlate it with the medical record. And there's a number of people looking at interesting things, such as if DNA is fragmented, broken into little things, does that predict that the cancer will be worse? Will that predict if the cancer will respond to a certain therapy? Will it tell us that it won't respond to something so we don't waste our time or cause excess toxicity? So this Moonshot program has garnered a lot of interest. It's one of the largest in the country, and we're very excited to see what it produces over the next few years. So what have we learned from clinical trials? We uncovered novel mechanisms of treatment. We're beginning to unravel novel mechanisms of disease so we can target things across different types. We're learning to offer therapies moving when we can offer these therapies, right? From stage 4 to pre-surgery, post-surgery, melanoma. Now we have neoadjuvant and adjuvant. We have immune therapies, colorectal, and we can move from specific cases to many different tumors. And what the idea is, we're trying to improve outcomes by screening as well. This is the most important thing. Diagnose cancer earlier when curable. And so I'm going to tell people to have their lung screening if you're a smoker, have your colonoscopy if you're age 45 or greater, have your mammogram, have your skin examined. And there's actually a very large there's no reason not to do it. Monthly across our network, we have at different places screening where you can go there and get screening for minimal or no fee. So if you don't want to get it out of your PCP, you can get it at AHN. So it's really important. The earlier we find it, the better we can treat it. So remember, we started off the what, when, and why. What is trials take many forms. Remember, we talked about looking back, retrospective trials, prospective, meaning going forward. Everybody gets the drug, randomized ones. These are expensive, they're time consuming, and they have to be timely. If you have a trial that takes 6 years and a year before it finishes, a new novel therapy comes up, you spent a lot of money. And so you want to do these effectively and efficiently. And when do you want to do these trials? You want to do it prophylactically. People are doing trials where they inject things into polyp areas when you have a colonoscopy, and can we prevent new polyps and therefore prevent cancer? When a cancer first appears in the first line of therapy or after failure, before an intervention, this thing we call neoadjuvant, after an intervention to reduce the risk of recurrence, when standard treatments are unavailable, or things like our moonshot, which are registry studies. Why? And why? Every cancer has to be studied in a trial. We will not move our needle anywhere if we don't have these and we don't have people participate. 20 to 40% of pediatric cancer patients participate in trials, but only about 1% of adult cancer patients do. Part of it's because they say they're busy, they don't want to, they don't want to be experimented on. But it's the only way we move the therapy, and we hope we can convince people that it's important. And it is the scientific method. It is this method that has brought every single life-saving treatment to medical care. Everything from cardiac to brain to strokes, everything that's gone in for vaccines for common childhood diseases, as well as things like HPV to reduce cervical cancer. All of these things had to go through trials to prove that they're effective and safe before we could give them. So with that, I'm glad to entertain questions and acknowledge the most important people who are their patients and their families who participated in these trials. So thank you very much for your attention, and I'm glad to discuss whatever it is you like. Are there questions, Michelle?
Don't see any questions.
Okay. I didn't think I was that clear, but okay, I'm glad. There'll be a quiz at the end.
Yeah, no questions so far.
Okay.
Let me just check one more time. No, we don't have any questions tonight.
Okay.
Okay,
Well,
well,
thank you all very much. I very much appreciate. Oh, there's a question, something in here.
oh, okay, I will, let me just,
Are all
are all
cancers
can,
diagnosed at AHN looked at for clinical trials? Yes. Um, I can't tell you specifically what is available for a stage 1B vulvar cancer. That would be something that we would talk to our GYN oncologist by. I don't have the whole list here. Non-HPV related. Yes. So what I would do is definitely, definitely would talk to the gynecologists who are doing this. They would know the answer if that specific stages. I apologize, you missed most of the presentation. Yes, it is available. Michelle, how is it available and where?
so it is, this evening's lecture was recorded. The video
Yeah.
will be available on the AHN YouTube channel
Yeah.
in about a week under the Ahead of Cancer playlist.
Right.
You can also find our past recordings there as well on YouTube.
Yeah.
And we'll show that message at the end as well. We'll show it.
So we got one from Robert Mertz. Is there a tool to screen patients' records with all available clinical trials to see if there is something promising? So across the nation, there is not a tool. So I can't put in your specifics or somebody's specifics and ask, is there a trial? And even if there is, so if you go to clinicaltrials.gov, you can almost always find clinical trials. And then you look, and they might be open in 9 places, but they may not all have openings. And you have to learn if you are actually available for it. But here at AHN, what we are doing is sticking as we're growing our clinical trials program, we've got a pathway system. So if you have, for instance, a stage 2 colon cancer, when we input it into there, if there's a stage 2 colon cancer trial, it will pop up for your doctor first. And so that's the way we're trying to ensure that people at least know about these across the network. And we're actually opening up, I didn't get time to really talk about it. We're opening up a thing with Tempest, which is a company where they have about 60 trials that are just in time that will be screened for all of our patients, and that we can open up in a couple of weeks if we identify a patient who's eligible. So probably by the first quarter of next year, that's March, April, we'll be able to screen for many more trials. And are there any trials for ovarian cancer? I happen to know there are, but I can't tell you which stages again. You will be able to answer in brain cancer. So about 2 years ago, we were lucky enough to bring on John Herbst, who is a neuro-oncologist from Memorial Sloan Kettering trained and from around these parts. And he's opened up a number of trials for glioblastoma and other cancers.
Okay.
Excision
Okay, we
and
have two
radiation
more.
already done, standard of care applied, clinical trials not mentioned. They may not mention if there's not one, but you can specifically ask your oncologist if there is one. I know that they've opened 3 in the last 2 months. When you're participating in trials, your treatment paid for by the grant. So anything that is not standard of care is paid for by the grant. So for instance, if the standard of care in an ovarian cancer trial is to do a CT scan every 2 months, and um, and we want to and for some reason they want to look at every 6 months, or if it's 3 months and they want to look at every 2 months, right? Then they would have to pay for the extra scan. If there's labs, they pay for the extra, but otherwise your insurance has to pay for it. Is the DNA of all cancers at AHN routinely sequenced? We're trying we now have a system in place. About 60%, it's really sort of insurance dependent upon the stage. So some insurances will only let us do it in stage 4 cancers or certain stage 3 cancers. If we can get them to pay for our internal panel, we can do it in a far wider range of patients, but some of that is unfortunately dictated by insurance companies and not us. Trials for prostate cancer stage 4. I happen to know that there are because our fellows were in a special conference this morning. Somebody presented a prostate cancer, and our 2 GU lead oncologists, Stefan Mao and Alexander Helfand, mentioned at least one. I think they said there were 2 trials open up in that area. But it sort of depends. Remember, a trial might be for people who've never been treated before, people who failed one line of chemotherapy. So it would really be specific for you. For brain cancer, John Herbst, Herbst.
Okay,
Really a gem.
I think we just ran over. I think we just ran over a few minutes, so I just want to conclude by thanking everybody for joining us for tonight's Ahead of Lecture Cancer Series. We hope you found it informative. Please join us for future lectures in our monthly series focusing on cancer screening, early detection, treatment, and survivorship. This evening's lecture was recorded, as we had said before, and it will be posted up on YouTube. If you have any questions regarding a potential cancer concern and would like to schedule an appointment, please call our Hope line at 412-578-4673. Thank you again for attending, and thank you, Dr. Bahary, for the excellent presentation. We hope you all have a pleasant evening.